Welcome to For Science Salah Lotfy channel, where science meets curiosity. في هذه القناة لا نكتفي بعرض الأبحاث العلمية، بل نترجمها إلى المعرفة التي تصل إليك أينما كنت. Our mission is to make global scientific research understandable, accessible, and multilingual. جميع الفيديوهات العلمية هنا مترجمة إلى اللغة العربية، بالإضافة إلى لغات عالمية أخرى. French, Spanish, German, Urdu, and more. Because science speaks every language. نعم، العلم يتحدث بكل اللغات، ونحن نحرص أن يصلك بلغتك. If you're passionate about science, you're in the right place. اشترك الآن وابدأ رحلتك مع العلم خطوة بخطوة. For Science Salah Lotfy channel, العلم للجميع. Welcome to For Science Salah Lotfy channel. Today I'm diving into a game-changing breakthrough led by Egyptian researchers. This is a story of innovation and dedication. They are right at the cutting edge of nanomedicine and radiopharmaceuticals, pushing the boundaries of what's possible. Let's talk about a smart, self-assembled nano platform for targeted cancer therapy. It's a revolutionary approach. Imagine this: magnetic iron oxide nanoparticles, tiny but powerful tools. They are combined with natural tannic acid, a compound derived from plants. And then there's the well-known chemotherapy drug doxorubicin, a staple in cancer treatment. All these elements are coming together, then radio-labeled with technetium-99m, making them traceable. This system isn't just about attacking tumors, it's about precision and control. It's about seeing and tracking them in real time, thanks to advanced nuclear imaging, providing invaluable insights. So what's the big vision behind this research? It's ambitious, a multi-pronged approach to revolutionize cancer treatment. First, combine chemotherapy and chemodynamic therapy in one powerful platform, maximizing the therapeutic effect. Second, enhance how precisely we can target cancer cells, ensuring the treatment reaches its intended destination. Third, cut down on the toxic effects healthy tissues suffer, minimizing side effects for patients. And finally, add real-time tumor imaging using radioactive markers, allowing for immediate assessment of treatment effectiveness. It's therapy and diagnostics in one theranostics, a truly integrated approach to medicine. Now, what did the scientists actually find? What were the tangible results of their hard work? They managed to successfully synthesize and self-assemble these nanoparticles, a crucial first step. More importantly, the particles homed in on cancer cells, leaving healthy tissue nearly untouched, a testament to their precision. Drug delivery was more effective, drugs stuck around in tumors longer, maximizing their impact, and crucially, the combined approach meant tumors were destroyed more efficiently than before, leading to better outcomes. Why does this matter to the scientific and medical world? What are the broader implications of this research? First, it's a leap for green nanomedicine, using natural compounds like tannic acid, promoting sustainability. On the technology front, it points toward multifunctional drug delivery systems, paving the way for more advanced treatments. Medically, it's a potential new hope for treating tough, drug-resistant cancers, offering a lifeline to patients in need. It also allows for seeing tumors with more precision, leading to more accurate diagnoses and treatment plans. But the impact doesn't stop at the lab. It extends far beyond the walls of the research facility. Economically, this could lower healthcare costs by making treatments faster and safer, benefiting both patients and healthcare systems. Because it uses accessible materials, there's a real chance for local production, creating jobs and boosting the economy. This is helping Egypt and the Arab world lead in this advanced field, fostering innovation and scientific advancement in the region. And socially, it's about hope. Hope for cancer patients and for communities everywhere, offering a brighter future. This research stands as proof. Egyptian scientists are innovating, and their ideas are changing lives, one breakthrough at a time. Local discoveries like this can create global waves, transforming science, the economy, and real people's futures, impacting lives worldwide. Subscribe to For Science Salah Lotfy channel for more stories where science empowers lives and knowledge transforms the world. Because science isn't just facts, it's a tool for change, a catalyst for progress, and a beacon of hope for a better tomorrow.
Welcome to For Science Salah Lotfy channel, your go-to source for exploring the fascinating world of scientific discovery, where science meets vision and discovery becomes a story worth sharing. We delve into the groundbreaking work of researchers who are shaping the future. Today, I'm shining a spotlight on one of the leading minds in radiopharmaceutical chemistry, a field that holds immense promise for the future of medicine. Professor Dr. Tamer Sakr, a name you'll want to remember. If you're passionate about the future of nuclear medicine and how it can revolutionize healthcare, targeted drug delivery, a method of getting medication directly to the source of the problem, or nanotechnology, the science of manipulating matter at an atomic and molecular scale, you're in for a treat. Prepare to be amazed by the scope and impact of his work. Dr. Sakr is a professor of radiopharmaceutical chemistry, dedicating his career to this vital area of research at the Egyptian Atomic Energy Authority, a leading institution in the region. He's also a technical consultant for radioisotope production, ensuring the safe and effective creation of these essential materials. And his expertise isn't limited to Egypt. His influence extends far beyond national borders. As an IAEA expert, working with the International Atomic Energy Agency, a Fulbright visiting scholar at the University of Missouri, fostering collaboration and knowledge exchange, and an academic at the National Defense College, contributing to strategic thinking and national security, Dr. Sakr's impact is truly global. He's a force for progress on an international scale. Now let's talk about his academic journey, the foundation upon which his remarkable career is built. Dr. Sakr earned his PhD in radiopharmaceutical chemistry, a testament to his dedication and intellectual rigor, from the Faculty of Pharmacy at Ain Shams University, one of Egypt's most prestigious institutions. His career bridges medicinal chemistry, the art and science of designing and developing new drugs, molecular modeling, using computer simulations to understand and predict the behavior of molecules, nanomedicine, applying nanotechnology to prevent and treat disease, and radiopharmacy, the specialized field of preparing and dispensing radioactive drugs. He's hands-on with radioisotopes, carefully working with these powerful tools of modern medicine, but he's also a master of advanced molecular imaging techniques, allowing us to see inside the body with unprecedented clarity. His research has sharpened drug formulation, making medications more effective and easier to administer, bioconjugation, linking biological molecules together for new applications, targeted radiotherapy, delivering radiation directly to cancer cells while sparing healthy tissue, and diagnostic imaging, providing crucial information for accurate diagnoses. What stands out is his commitment to making diagnosis and treatment, pushing the boundaries of what's possible in healthcare, more precise, tailoring treatments to the individual needs of each patient, less invasive, minimizing the impact on the patient's body and improving recovery times, and, most importantly, accessible to all, ensuring that everyone, regardless of their background, can benefit from these advancements. With over 100 peer-reviewed publications, Dr. Sakr has significantly contributed to the scientific community. Dr. Sakr's research pivots around 5 key themes. These are the pillars of his groundbreaking work. Let's break these down. We'll explore each theme in detail to understand their significance. First, radiopharmaceuticals for cancer diagnosis and therapy, a critical area in the fight against this devastating disease. Dr. Sakr has developed technetium-99m labeled probes, using this versatile radioisotope to create targeted imaging agents and radioiodinated compounds to image tumors, helping doctors to detect cancer earlier and more accurately. His work on 99mTc-DOX gold nanoparticles is a game-changer, combining the power of nanotechnology with targeted drug delivery. Imagine a single tool that diagnoses and treats cancer at the same time. This is the promise of theranostic medicine, and Dr. Sakr is leading the way. He's also worked on 99mTc-gemcitabine, exploring new ways to deliver this important chemotherapy drug, and radioiodinated doxorubicin, further refining our ability to target and destroy cancer cells, both pushing the boundaries of tumor imaging and therapy. These advancements offer hope for more effective and less toxic cancer treatments. Second, molecular modeling and targeted drug delivery. Using the power of computers to design better drugs and deliver them more effectively, Dr. Sakr uses in silico methods to design smarter drugs. This approach saves time and resources by predicting how drugs will behave before they are even synthesized. Computational modeling has led him to enhance radiolabeling, improving the process of attaching radioactive isotopes to molecules, for molecules like oxiracetam and tanoxicam, optimizing their properties for imaging and therapy, making cancer cell targeting more accurate, ensuring that drugs reach their intended target with minimal side effects. He's even explored repositioning existing drugs, finding new uses for medications that are already approved for other conditions, like phosphonothrycin, for new imaging purposes. This innovative approach can accelerate the development of new diagnostic tools. Third, nanotechnology and nanomedicine, harnessing the power of the very small to revolutionize healthcare. Dr. Sakr is at the forefront of combining nanomaterials with radiopharmaceuticals, creating new possibilities for targeted drug delivery and imaging. He's synthesized selenium nanoparticles, exploring their potential for cancer therapy and imaging, gold nanostructures, utilizing their unique properties for targeted drug delivery and photothermal therapy, and graphene oxide-based probes, developing new tools for imaging and treating cancer at the molecular level, all for advanced cancer therapy and diagnostics. These innovations promise to improve the lives of countless patients. Vitamin C-stabilized selenium nanoparticles for tumor imaging, a novel approach that enhances biocompatibility and targeting. That's just one of his many breakthroughs. Dr. Sakr's creativity and ingenuity are constantly pushing the boundaries of what's possible. Fourth, innovative imaging agents for non-cancer applications, expanding the reach of nuclear medicine beyond oncology. Dr. Sakr's research isn't only about cancer. He's dedicated to improving the diagnosis and treatment of a wide range of diseases. He's developed agents for heart imaging, helping doctors to diagnose and manage cardiovascular disease, lung scintigraphy, providing valuable information about lung function and respiratory disorders, and infection detection, enabling earlier and more accurate diagnosis of infectious diseases. Like 99mTc-lidocaine for cardiac imaging, a novel approach to assessing heart function, and ceftriaxone radio-labeling for pinpointing infections, helping to guide antibiotic therapy and improve patient outcomes. Fifth, contributions to global radiopharmacy and policy, shaping the future of nuclear medicine on a global scale. Dr. Sakr is a key player, actively involved in shaping the policies and guidelines that govern radiopharmacy worldwide, in advancing nuclear medicine infrastructure worldwide, working to ensure that these life-saving technologies are available to all. His reviews and editorials shape the direction of the field, providing expert guidance and insights to the scientific community. From the IAEA's roadmap for nano-sized radiopharmaceuticals, setting the stage for future innovations in this exciting area, to analyses of radiopharmacy in Africa, addressing the unique challenges and opportunities for nuclear medicine in this region. What ties all this together is Dr. Sakr's vision for precision medicine, a future where treatments are tailored to the individual characteristics of each patient. He believes the future of therapy is personal, moving away from a one-size-fits-all approach to healthcare, guided by imaging, using advanced imaging techniques to monitor treatment response and adjust therapies accordingly, powered by nanotechnology, leveraging the unique properties of nanomaterials to deliver drugs and therapies with greater precision, and grounded in chemistry and biology. Integrating these fundamental sciences to develop innovative solutions to medical challenges. His collaborations span Africa, fostering scientific partnerships and building research capacity across the continent, the Middle East, promoting regional cooperation and addressing shared healthcare challenges, Europe, engaging in cutting-edge research and knowledge exchange with leading European institutions, and the U.S., collaborating with American researchers to advance the frontiers of radiopharmaceutical science, building a truly global network for scientific innovation, connecting researchers and institutions around the world to accelerate progress in healthcare. Through his research and partnerships, Dr. Sakr is leaving an indelible mark on the world of science and medicine. Professor Dr. Tamer Sakr is transforming radiopharmaceutical science, paving the way for a healthier future for all, making diagnostics smarter, enabling earlier and more accurate detection of disease, treatments safer, minimizing side effects and improving patient outcomes, and outcomes better for patients everywhere. His work is a testament to the power of science to improve human lives. This has been another spotlight from For Science Salah Lotfy channel, bringing you the stories of the scientists who are shaping our world. Where groundbreaking science meets storytelling. We hope you've enjoyed learning about the remarkable work of Dr. Tamer Sakr. Stay curious, and keep exploring the wonders of science with us. Hey there, and welcome to For Science Salah Lotfy channel, where scientific innovation meets real-world impact. Today I'm shining a spotlight on an incredible breakthrough in the world of cancer nanomedicine, coming straight from Egyptian researchers who are truly at the cutting edge. Let's dive right in. The research we're exploring is titled An Intelligent and Self-Assembled Nanoscale Metal Organic Framework, 99mTc-DOX FeIII-TA acid for tumor-targeted chemodynamic theranostics. Try saying that 5 times fast. This study was published in Cancer Nanotechnology in 2024, Volume 15, Article number 28. It's a big leap forward in blending cancer therapy with nuclear medicine imaging, and it introduces a brand new, self-assembled nanoscale platform. Now let's meet the brilliant minds behind this research. First up, Dr. Mohamed Swidan from the Department of Labeled Compounds, Hot Lab Center at the Egyptian Atomic Energy Authority in Cairo. He's also the corresponding author, so if you've got questions, he's your go-to guy. Next, Dr. Nehal Wahba, hailing from the Department of Pharmacology and Toxicology at the Faculty of Pharmacy, Zagazig University. Her expertise in pharmacology was crucial for making sure this nano platform is both effective and safe. And finally, Professor Dr. Tamer Sakr from the Radioactive Isotopes and Generators Department, also at the Hot Lab Center, Egyptian Atomic Energy Authority. He's the radiopharmaceutical chemistry expert who made the nuclear medicine side of this project possible. So, what's this research actually about? The team engineered a smart, self-assembling nanoscale metal organic framework. Here's the recipe: iron oxide nanoparticles, functionalized with tannic acid, loaded with the chemo drug doxorubicin, and radio-labeled with technetium-99m. This powerhouse combo brings together three big things: chemotherapy, to attack and destroy cancer cells directly; chemodynamic therapy, which ramps up oxidative stress inside tumors; and SPECT imaging, where the radioactive label lets doctors track where the particles go in the body. All in one. That's what makes theranostics so exciting. Now, why does this matter? This work shows how Egyptian scientists are pushing the limits of what's possible in targeted cancer therapy by blending nanotech, nuclear chemistry, and pharmacology. The potential here? More accurate treatments, fewer side effects, and lower healthcare costs, not just in Egypt but across the whole region. It's more than a scientific paper. It's a vision for a healthier future. If you want to stay up to date with more research breakthroughs from Egypt and the Arab world, make sure you like, share, and subscribe to For Science Salah Lotfy channel. Science belongs to everyone. Thanks for watching.
Hey science lovers, welcome to For Science Salah Lotfy channel. Today I'm sharing a breakthrough from Egyptian scientists that could seriously change how we fight cancer by blending nanotechnology, chemotherapy, and nuclear imaging into one powerful weapon. This research comes from a team led by Dr. Mohamed Swidan, Dr. Nehal Wahba, and Professor Dr. Tamer Sakr. Together, they created what's basically an intelligent, self-assembled nanomachine designed to target and destroy tumors, while letting doctors see exactly what's happening inside the body. Alright, let's start with the problem. Cancer kills millions every year. Chemotherapy, especially with drugs like doxorubicin, can be effective, but it also causes harsh side effects. And while chemodynamic therapy, or CDT, has promise, it faces three big challenges. There isn't enough hydrogen peroxide in tumors, the tumor environment isn't acidic enough, and there's lots of glutathione, a molecule that actually protects the tumor. That's like fighting with your hands tied. So what's the solution? The team engineered a smart nano platform with a pretty wild name: 99mTc-DOX loaded AA-Fe₃O₄ FeIII-TA acid. Think of it like a supercharged delivery truck. It's got an iron oxide core coated with ascorbic acid, a shell made of tannic acid and ferric ions, and it's packed with doxorubicin, the chemo drug. Plus, it's labeled with technetium-99m, a radioactive tracer doctors use for nuclear imaging. Here's how it works. In your bloodstream, this nano truck stays locked up tight, no leaking cargo. But as soon as it finds itself in the acidic environment of a tumor, the shell breaks apart. Doxorubicin is released to attack the tumor DNA, tannic acid and ferric ions go to work, and here's the twist inside the tumor. Ferric ions react with glutathione, which not only wipes out the tumor's defenses, but also triggers the Fenton reaction. That reaction produces hydroxyl radicals, tiny but deadly molecules that obliterate cancer cells from the inside. Meanwhile, the technetium-99m lets doctors watch the whole process using real-time SPECT imaging. It's chemo, chemodynamic therapy, and diagnostics all rolled into one. So, did it actually work? Lab results showed almost perfect precision at the tumor's pH. 98% of the drug is released while at normal pH. Only 10% leaks out. It's highly stable in the bloodstream, finds its way to tumors, and lights them up for imaging. 16.8% of the injected dose per gram in the tumor just an hour after injection. And get this, the drug is 8 times more likely to target the tumor than healthy tissue. That's some next-level accuracy. To sum it up, this Egyptian-made nano-platform combines targeted chemo, enhanced chemodynamic therapy, and real-time imaging. It only activates where it's needed and goes after tumors with triple force. Smart, safe, and effective. It's a leap toward personalized cancer treatment, showing the global power of science right here from Egypt. Thanks for watching. If you believe science can heal and transform, hit subscribe and join for Science Salah Lotfy channel, where research becomes hope.
Today we're diving into a real-life scientific quest, building a microscopic robot to fight cancer. A team of scientists in Egypt faced a big question. What materials could make a safe, effective cancer-fighting nano-platform? Their answer started with copper sulfide, a material that heats up under special light to cook cancer cells from the inside out, without harming healthy tissue. Plus, copper ions can trigger chemical reactions in the acidic environment of tumors, making it a double threat. But copper sulfide alone isn't enough. To keep these particles stable and safe in the body, they wrap them in polyvinyl pyrrolidone or PVP, a kind of cozy sweater that prevents clumping and helps the particles move stealthily through the bloodstream. Next, they needed a smart shell to carry and release a cancer drug only at the tumor site. They built a metal organic framework, or MOF, using zinc ions and 2-methylimidazole. This MOF forms a porous, cage-like shell around the core, stable in the bloodstream but designed to break down in the acidic environment of tumors. That pH sensitivity is the secret switch, ensuring the drug is released only where it's needed. So, the nano platform is a three-part superhero: a copper sulfide core for heat and chemistry, a PVP sweater for stability, and a MOF shell for smart drug delivery. It's a tiny targeted cancer-fighting machine. This clever design is the foundation for everything that follows. Let's see how they put it all together. The adventure begins. Building this nano platform started with making the copper sulfide core tiny crystals formed by mixing copper salt and sulfur in a controlled reaction. Next, the scientists coated these particles with PVP, making them stable and water-soluble for safe travel in the body. Then came the smart shell. They placed the PVP-coated cores in a solution with zinc ions and 2-methylimidazole. These components self-assembled into a MOF shell, creating a porous cage around the core. The real genius? The MOF shell stays intact in healthy tissue but breaks down in acidic tumor environments. This pH-triggered breakdown is the key to targeted drug delivery. The result, a stable, smart core shell nanoplatform ready to carry medicine straight to cancer cells. Every step was about precision, making sure the platform was the right size, shape, and stability for its mission. With the chassis built, it was time to load it with a weapon. The stage was set for a new kind of cancer therapy. Let's see what happens next. Now the scientists loaded their nano-platform with doxorubicin, a powerful chemotherapy drug. Thanks to the MOF's porous structure, the drug soaked in easily, like water into a sponge. They tested the release in neutral conditions. The drug stayed put. In acidic, tumor-like conditions, the MOF shell broke down and released the drug right where it was needed. But this platform had another trick: chemodynamic therapy. In tumors, copper ions from the core react with hydrogen peroxide to create destructive hydroxyl radicals, molecules that attack cancer from within. The team confirmed this by mixing their nano-platform with hydrogen peroxide and a special dye. The color change proved radical production. So this isn't just a drug delivery system, it's a two-pronged attack-targeted chemotherapy and radical generation. The nano-platform is ready to unleash its full power on cancer cells, smart, targeted, and deadly to tumors. The next step: real-world testing. Could it work on actual cancer cells? The answer was about to be revealed. Let's find out. The scientists tested their nano-platform on human breast cancer cells in the lab. Compared to regular chemotherapy, the smart nano-platform killed far more cancer cells, especially when activated by light. The secret? A powerful combination: drug release, photothermal heat, and radical generation. Under the microscope, treated cells glowed green, showing a flood of destructive reactive oxygen species. The platform didn't just attack, it also depleted the cell's main defense, glutathione, leaving them defenseless. The result: cancer cells overwhelmed and destroyed. This was proof the nano-platform worked as designed, a major victory in the fight against cancer. But could it work in a living body? The ultimate test awaited. Next, the team tested their nano-platform in mice with human breast cancer tumors. To track the particles, they attached a radioactive label, making them visible to medical imaging. After injection, the nano-platforms homed in on the tumors, confirmed by a bright signal on the gamma camera. This showed the platform's targeting power in action. For treatment, some mice received the full nano-platform system, including light activation. The results were dramatic. Tumors shrank significantly, and some disappeared entirely. This was the ultimate proof of concept. Targeted, multi-pronged therapy delivered by one smart nanoplatform. The research offers a blueprint for future cancer treatments, smarter, safer, and more effective. Imagine the possibilities. Science is turning tiny technology into big hope. The future of cancer therapy is here. Have you ever stopped to consider why treating cancer remains such a formidable challenge, even with all the advancements in modern medicine? Part of the answer, a significant part, lies in the astonishing resourcefulness of cancer cells. They are incredibly clever masters of adaptation, especially when it comes to protecting themselves from our most advanced and potent medicines. It's a constant arms race, a battle of wits and chemistry. One of the key building blocks, a fundamental component of this protective fortress, is a fascinating molecule called glutathione, often abbreviated as GSH. At its core, glutathione is a powerful antioxidant. This means it has the remarkable ability to neutralize harmful substances within the body, acting like a tiny cleanup crew, scavenging and eliminating threats. In a healthy body, the antioxidant properties of glutathione are undeniably a good thing. It supports cellular health and overall well-being. But in the complex and often perverse world of cancer, this normally helpful molecule takes on a villainous new role. It gets co-opted, hijacked, and transformed into the cancer cell's personal bodyguard, an essential part of its survival strategy. Imagine trying to fight a formidable villain who possesses an invisible, almost impenetrable shield. That's precisely what many cancer therapies, including the widely used chemotherapy and radiation, are up against when battling cancer cells. It's like trying to break through a fortress with conventional weapons. These treatments, chemotherapy and radiation, work by creating oxidative stress within the cancer cells, essentially overwhelming them with harmful free radicals in an attempt to destroy them. But the cancer cells, ever resourceful, are ready for this attack. They cleverly crank up their GSH production, synthesizing more and more of this protective molecule to neutralize our treatments and blunt their effectiveness. It's a sophisticated defense mechanism at play. As a result, the intended therapy becomes significantly less effective than hoped. Cancer cells manage to survive the onslaught and, even worse, continue to spread and proliferate, undermining the entire treatment strategy. This sophisticated defense mechanism, orchestrated by glutathione, represents one of the most significant and persistent challenges in modern oncology, demanding innovative solutions and novel therapeutic approaches. Overcoming this hurdle is crucial in the ongoing fight against cancer. To solve a big problem, sometimes you have to think very, very small. In the world of cancer research, scientists are constantly seeking innovative solutions, pushing the boundaries of what's possible. One such breakthrough involves shrinking the battlefield down to the nanoscale. Researchers have developed an incredible new tool called a nano-metal organic framework, or Nano-MOF for short. These aren't your average molecules, they're meticulously designed structures with a specific mission. Think of it as a microscopic, super smart sponge engineered for a targeted attack on cancer cells. They are so small you could fit millions of them on the head of a pin. To give you a sense of scale, imagine covering the period at the end of this sentence with these nano-MOFs. You could pack thousands onto that tiny dot. But don't let their size fool you. These miniature marvels pack a powerful punch. These little structures are designed with a very specific purpose, to seek out and deplete the cancer's glutathione shield. Glutathione, or GSH, is a molecule that cancer cells use to protect themselves from harm. It acts like a bodyguard, neutralizing threats and allowing the cancer to thrive. The Nano-MOF is designed to disarm this bodyguard, leaving the cancer vulnerable. The Nano-MOF was engineered to have high affinity for glutathione. It's like a lock and key, where the Nano-MOF is the key specifically designed to fit the glutathione lock. When it contacts a cancer cell packed with GSH, it acts like a magnet, pulling glutathione molecules out. The interaction is swift and precise, a testament to the careful engineering behind these nanostructures. It traps them within its porous structure. Imagine a tiny cage, perfectly sized to capture and hold the glutathione molecules. This process is called GSH depletion. By soaking up the glutathione, the Nano-MOF dismantles the cancer cell's primary defense system. It's a strategic maneuver, weakening the enemy before launching a full-scale attack. It leaves the cell exposed and unable to protect itself from oxidative stress that our treatments deliver. Without its glutathione shield, the cancer cell is like a soldier without armor, vulnerable to the weapons of modern medicine. It's like flipping a switch, turning the cell's environment against it, making it far more susceptible to the effects of chemotherapy and radiation. This innovative approach holds immense promise for improving cancer treatment outcomes. Creating a tiny invisible weapon is great, but how do doctors know if it's working? How can they see where it's going inside the body? The researchers found a way to make their Nano-MOF visible to medical imaging equipment through radio labeling. This tag is Technetium-99em, one of the safest radio tracers in medicine. Think of it as a tiny light bulb screwed into our Nano-MOF. This turns it into a theranostic agent, combining therapeutic and diagnostic functions in one tool. When this radio-labeled Nano-MOF is introduced into the body, doctors can follow its journey in real time, watching as it travels through the bloodstream. They can see exactly where it accumulates and confirm that the medicine is going directly to the tumor, not wandering off to other parts of the body. The next crucial step was testing this radio-labeled Nano-MOF in living systems. Scientists conducted preclinical studies using mice with tumors, essential for understanding biological behavior. The results were remarkable. The studies showed that the Technetium-99m tag stayed securely attached as it traveled through the bloodstream. This high stability is crucial for accurate tracking. Most importantly, the imaging showed clear results. The radio-labeled nano-MOFs were accumulating significantly in the tumors, actively congregating at cancer sites. The scans lit up right where the tumors were, showing high nano-MOF concentration. This excellent targeting means the medicine gets to the right place to do its job. This successful demonstration is a major milestone, providing strong evidence that this system works in living organisms, paving the way for future human applications. What does this mean for the future of cancer treatment? We're standing on the precipice of a paradigm shift, a fundamental change in how we approach this disease. This Nano-MOF, this tiny yet powerful innovation, represents a whole new strategy, one that aims to make cancer treatment not just more effective but also more intelligent. It's about targeting the disease with precision, minimizing harm to healthy cells, and ultimately, improving the lives of patients. Imagine a future where chemotherapy is a thing of the past, replaced by therapies that are tailored to each individual's unique cancer profile. This is the promise of Nano-MOFs. This innovative see-and-treat approach allows doctors to personalize care in unprecedented ways. Before administering powerful therapies, they can confirm that the drugs are precisely on target, hitting the cancerous cells while sparing healthy tissue. This maximizes effectiveness, ensuring that the treatment is working as intended, while simultaneously minimizing side effects, reducing the burden on the patient's body. It's about delivering the right drug, to the right place, at the right time, all guided by advanced imaging and personalized treatment planning. This level of precision is a game changer in the fight against cancer. It's a smarter, more targeted way to fight cancer, and it has the potential to change outcomes for millions of people around the world. The significance of this research simply cannot be overstated. It represents a beacon of hope for patients and families affected by this devastating disease. This isn't just about incremental improvements. It's about a fundamental shift in how we think about and treat cancer, offering the promise of longer, healthier lives for countless individuals. The impact could be felt globally, transforming healthcare systems and improving the quality of life for people everywhere. On behalf of Professor Dr. Salah Lotfy and our entire channel, we extend our deepest gratitude and sincere appreciation to the brilliant minds behind this groundbreaking work. We want to especially thank Dr. Mohamed Swidan, Dr. Nehal Wahba, and Professor Dr. Tamer Sakr for their tireless efforts and unwavering commitment to advancing the field of cancer research. Their expertise and dedication have paved the way for this incredible breakthrough. Their unwavering dedication, relentless pursuit of knowledge, and remarkable ingenuity have given us a powerful new tool in the fight against cancer and renewed hope for a future free from this disease. This is what science is all about, pushing the boundaries of what's possible, challenging conventional wisdom, and striving to make the world a better place for all. It's about collaboration, innovation, and a shared commitment to improving human health. Even with the presence of high levels of reactive oxygen species, or ROS, which are naturally toxic to cells, tumors have developed a clever defense mechanism to ensure their survival. This defense primarily revolves around a molecule called glutathione, often abbreviated as GSH. Think of it as an antioxidant shield, meticulously protecting cancer cells from the constant threat of oxidative attacks. It neutralizes the harmful ROS, preventing them from damaging critical cellular components and allowing the cancer to thrive. So what if we could find a way to weaken that shield, to disrupt this protective mechanism that glutathione provides? What if we could leave the cancer cells vulnerable to the very ROS that should be destroying them? This is a critical question in cancer research. That's exactly what this smart Nano-MOF platform aims to achieve. This innovative approach targets the glutathione defense system directly. The research team discovered that cancer cells treated with their specifically engineered Nano-MOF experienced a significant reduction in GSH levels. This means the protective shield was effectively being dismantled by the Nano-MOF. The experiments showed GSH levels plummeting down to 47% of their original concentration, and even further, down to a mere 32% when the Nano-MOF was loaded with the chemotherapy drug DOX, or doxorubicin. This dual effect is key. It not only strips away the cancer cell's glutathione defenses, making them vulnerable, but also simultaneously boosts the production of ROS, amplifying the oxidative stress and pushing the cancer cells closer to self-destruction. To visualize and track the delivery of our drug, we employed a sophisticated technique called radiolabeling. This involved tagging the DOX, or doxorubicin, with technetium-99em, a radioisotope widely used in medical imaging. The process was meticulously optimized to ensure that the technetium-99em was securely attached to the DOX molecule, allowing us to monitor its journey within the body. This radiolabeling process achieved an impressive 96% radiolabeling efficiency, meaning that almost all of the DOX molecules were successfully tagged with the radioisotope. Furthermore, we achieved 98% loading of this radiolabeled DOX onto the Nano-MOF, ensuring a high concentration of the drug within our delivery vehicle. Maintaining stability in biological environments is crucial for any drug delivery system. To assess this, we subjected our system to rigorous testing in mouse serum, a complex fluid that mimics the conditions within the bloodstream. The results were highly encouraging. The system maintained over 94% stability for a period of 8 hours in mouse serum. This remarkable stability suggests that our Nano-MOF is robust enough to withstand the harsh conditions within the body, protecting the drug from premature degradation and ensuring that it reaches its target. This strong stability shows significant promise for real-time tracking and effective drug delivery in vivo. To evaluate the effectiveness of our targeting strategy, we conducted in vivo imaging studies using tumor-bearing mice. These studies allowed us to visualize the distribution of the Nano-MOF within the body and to assess its accumulation in tumor tissue. Using SPECT imaging, we observed a significant accumulation of the Nano-MOF in the tumor. In tumor-bearing mice, the system showed 16.8% tumor uptake at just 1 hour post-injection. This level of accumulation is remarkable, especially considering the short time frame. Even more impressively, this uptake was over 11 times higher than that observed with naked DOX alone, highlighting the superior targeting capabilities of our Nano-MOF delivery system. To further quantify the specificity of our targeting, we calculated the tumor-to-muscle ratio, which compares the amount of Nano-MOF accumulating in the tumor to that accumulating in surrounding muscle tissue. A high tumor-to-muscle ratio indicates that the Nano-MOF is selectively targeting the tumor while sparing healthy tissues. The tumor-to-muscle ratio reached an impressive 8:1, indicating precise tumor targeting. This high ratio strongly suggests that our Nano-MOF is effectively exploiting the enhanced permeability and retention (EPR) effect, a phenomenon where nanoparticles preferentially accumulate in tumor tissue due to its leaky vasculature. Imagine a future where cancer treatment is not just about fighting the disease, but also about understanding it at its most fundamental level. This nano platform represents a significant leap towards that future. It's not just a treatment, it's a sophisticated diagnostic and therapeutic system ingeniously integrated into one. This truly intelligent platform is designed to be pH sensitive, reacting to the subtle changes in acidity within the tumor environment. Furthermore, it's redox responsive, meaning it can detect and respond to the oxidation reduction processes that are characteristic of cancer cells. This dual sensitivity allows for targeted drug delivery and real-time monitoring of treatment effectiveness. This groundbreaking research is made possible with crucial support from the Science, Technology, and Innovation Funding Authority. Their commitment to fostering scientific advancement allows us to bridge the gap between innovative ideas and real-world impact. This funding enables researchers to push the boundaries of what's possible, accelerating the development of new cancer therapies and diagnostic tools. It's a testament to the power of collaboration and the importance of investing in scientific discovery for the benefit of society. The journey from initial in vitro results, demonstrating efficacy in controlled laboratory settings, to in vivo success, showcasing its effectiveness in living organisms, marks a significant milestone. This innovative nano-MOF system offers a promising new horizon in the field of precision cancer theranostics. By combining diagnostic and therapeutic capabilities at the nanoscale, we can achieve more targeted and effective treatments, minimizing side effects and improving patient outcomes. This represents a paradigm shift in how we approach cancer therapy, moving towards personalized medicine tailored to the individual characteristics of each patient's disease. Don't miss out on the latest breakthroughs in science and technology. Subscribe to For Science Salah Lotfy channel today for more in-depth insights into the cutting edge of scientific innovation. Stay informed about the groundbreaking research shaping our future and join our community of science enthusiasts. Hit that subscribe button and enable notifications so you never miss an update.
As we conclude this episode, I want to express my deepest appreciation for the exceptional scientific work that made this research possible. It's truly inspiring to witness such dedication and innovation in the field of tumor-targeted therapy. The collaborative spirit and the relentless pursuit of knowledge are what drive scientific progress forward. We are immensely grateful for the contributions of every individual involved in this groundbreaking research. Their efforts have not only expanded our understanding, but also paved the way for future advancements in radiopharmaceuticals and oncology. On behalf of Professor Dr. Salah Lotfy from the Egyptian Atomic Energy Authority and this channel, I sincerely thank Dr. Mohamed Swidan, Dr. Nehal Wahba, and Professor Dr. Tamer Sakr. Your expertise and commitment have been instrumental in achieving these remarkable results. We deeply value your contributions and the collaborative spirit you brought to this project. Your willingness to share your knowledge and insights has enriched our understanding and propelled the research forward. We extend our heartfelt gratitude for your unwavering support and dedication. Your dedication has advanced our understanding of tumor-targeted therapy and raised the bar for interdisciplinary collaboration in radiopharmaceuticals and oncology. The impact of your work extends far beyond the laboratory, offering hope for improved cancer treatments and a better quality of life for patients. Your commitment to excellence serves as an inspiration to researchers around the world. We commend your passion for scientific discovery and your unwavering pursuit of innovative solutions. Your contributions have significantly advanced the field and set a new standard for collaborative research. To our viewers and scientific enthusiasts, thank you for joining us on this journey of discovery. We hope this episode has sparked your curiosity and inspired you to learn more about the fascinating world of radiopharmaceuticals and oncology. Your engagement and support are invaluable to us. We encourage you to continue exploring the wonders of science and to embrace the power of knowledge. Share your thoughts in the comments below. We are eager to hear your perspectives and insights on this important topic. Let's continue the conversation and work together to advance scientific understanding. Stay tuned to For Science Salah Lotfy channel, where we bring you the latest scientific innovations from around the world. We are committed to providing you with informative and engaging content that explores the frontiers of scientific discovery. Our goal is to inspire a passion for science and to empower you with the knowledge to make informed decisions. Until next time, keep questioning, keep exploring, and keep believing in the power of science. The pursuit of knowledge is a lifelong journey, and we are honored to be a part of it with you. Together, we can unlock the secrets of the universe and create a better future for all.